In this video, we're going to have a look at Module 612, specifically the patterns of inheritance that students regularly get mixed up, which include the difference between dihybrid inheritance and autosomal linkage. Sex-linked diseases will be covered in a different video. Let's start with a recap of monohybrid inheritance, which was first covered for GCSE biology, which is when you are looking at the inheritance of a single gene-controlled characteristic. So, for example, our eye colour is controlled by one gene. The colour of peas is controlled by a single gene. The example on the screen now is looking at the inheritance of different shaped wings in fruit flies. The genotype for having long wings is shown, and it's represented with two capital N's. That means that this individual is homozygous dominant. Homozygous because both versions of that gene that they have inherited from their parents are the same as each other, and you can tell it's dominant because it's represented by a capital letter, and that is the typical convention, to show a dominant characteristic with a capital letter and a recessive one with a lowercase letter. Vestigial wings are shorter, they have a slightly different shape, and you can see that the genotype for vestigial wings is two lowercase n's. This individual is homozygous recessive. They have two alleles for the same gene which are the same as each other. They are both recessive, which is why this individual has vestigial wings. If they were heterozygous, i.e. they had one big N and one little n, the large N is the one that would be expressed, so that individual would have long wings, but they would still be carrying the allele for the recessive vestigial wing. The expression of that vestigial wing allele is going to be masked, hidden, by the fact that the long-winged allele is the one that is actually being expressed, but they can still pass on their recessive allele to their offspring during the process of meiosis when they produce their gametes. When we work out a monohybrid inheritance cross, typically it's the most straightforward thing to represent that cross by using a Punnett square. Each parent's gametes are presented either along the column headings or the row headings. So if we take the long-winged one as one of the parents, when meiosis is carried out, their genotype, their combination of alleles, is going to be split up so that one of those ends is going to be in one set of gametes and the other one is going to be in another. So they have made haploid cells which only have half the number of chromosomes as the original parent's genotype, and that's how they've been divided up. For the vestigial wing parent, when meiosis occurs and separates these two alleles, those two alleles are separated in this way. That's how we represent the parent's gametes. Now, what we do with a Punnett square is establish what the probability is of any one of these parents' gametes being fertilized by the other. So, for example, if this gamete was fertilized by this one, then the offspring would have a genotype which is heterozygous. It has one allele of each, and because long wings is dominant, this individual would have long wings. In fact, when you have this kind of a genetic cross between parents who are both homozygous, one homozygous dominant, one homozygous recessive, there is only one possible genotype for the offspring. 100% of the offspring would be heterozygous, which means 100% of the offspring would have long wings. This generation is what we call the F1 generation. And if you take two of these individuals and crossbreed them together, consider what the ratio of phenotypes would be with their offspring. Pause a moment and then check your answers on the next screen. For the F1 generation, both parents are heterozygous, so there's one parent's separation of the genotype to make the gametes, and here's the other parents. So when you put them together, this is the combination that you would expect. This individual is homozygous dominant, long wings. These two individuals are both heterozygous, long wings, and there is a 1 in 4 chance that the offspring would be homozygous recessive, so they would have short wings. The way we express that is often we would say 3 long wings to 1 short wing. You could also say 75% of the offspring are likely to have long wings and 25% of the offspring are likely to have short wings. When we take this up to the next level, we start looking at dihybrid inheritance, which is when you're looking at the inheritance of 2 characteristics controlled by different genes. One example that comes up regularly is looking at peas. This comes up because Gregor Mendel first did his genetics experiments on pea plants. They have a number of different characteristics which are all monogenic. They're all controlled by a single gene. And once you've established that, it makes it easier to make comparisons, to breed 2 parent plants together and see what the phenotypic ratios are in the next generation. Dihybrid inheritance means we don't just want to know what's the phenotypic ratio of colours that you might expect in the next generation, or what's the phenotypic ratio of the shape of seeds that you could expect in the next generation. You're combining those 2 characteristics. If these are the parents you have, what's the probability of having round and yellow seeds in the next generation, or wrinkled and yellow seeds? So you're looking at the inheritance of 2 different characteristics in the same individuals at the same time. In a diploid cell, a nucleus will contain 2 copies of every single gene, one on the maternal chromosome and one on the paternal chromosome. Those 2 genes may have different versions. For example, there might be one allele on one chromosome that says these peas should be round, and on the same homologous chromosome, the one belonging to the same pair, for the other parent, there might be in the same locus a gene that has the version, the allele, that says it should be wrinkled. In a gamete, that cell is haploid, which means it only contains one allele from every single gene. So instead of having 2 of each, round and wrinkled, it will only have one allele. However, the whole of the genetics within that gamete is still going to contain one version of every single gene. So within the gamete, it will have one allele for the shape and one allele for the colour. So when we separate the parent's genotype to identify which alleles are going to be in the gametes, we have to make sure that every gamete contains one of each characteristic. Let's do a worked example between 2 parents, one who is homozygous dominant for both characteristics, and one parent who is homozygous recessive for both characteristics. The parent who is homozygous dominant for both characteristics is going to have round, yellow seeds. The parent who is homozygous recessive for both characteristics is going to have wrinkled, green seeds. And I worked that out based on the key that I've been given in the original information. Step 1 for working out a dihybrid inheritance cross is always going to be to establish the genotypes and the phenotypes of the parents and make a note of that. Step 2 is going to be to establish which combination of these alleles are going to be in the gametes produced from each parent. Let's use the homozygous dominant parent as our first example. When meiosis occurs, the 2 capital R's, which are each on separate chromosomes but within the same pair, are going to be split up. So the gamete will either have a capital R or it will have the other capital R. Okay? There isn't anything else it can possibly have. The 2 Y's for the colour are also going to be separated out. So the gametes will either have a capital Y or they will have a capital Y, i.e. there's no difference, which means regardless of which of these alleles are passed on, they will always have the same colouring alleles attached to them within that gamete. What this looks like in a Punnett square, we would draw the Punnett square like this. That is the only thing that that particular parent can pass on. For the other parent who was homozygous recessive, they have to have a little r in each of their gametes. And because the colouring is also recessive, that's the only combination they can possibly pass on. There is nothing else. So when we draw out our lines and you show what the possible fertilised offspring are going to have, it's important to combine both sets of alleles from each gamete. When this individual is fertilised by this individual, they will have one big R and one little r, one big Y and one little y, i.e. they are now going to be heterozygous for both characteristics. Because round is dominant, they are going to have round seeds. Yellow is dominant, so they are all going to have yellow seeds. And so you're actually going to end up, when you work this out for all of the 4 possible combinations, with 100% of individuals who have round yellow seeds. There is nothing else you can possibly get. So step 3 is looking at working out what the possible genotypes are when these cells are fertilized. And step 4 is then establishing for each genotype what is the offspring genotypes, and then what are the percentage ratios of the phenotypes. Similar to when we had a look at the fruit flies, this is the F1 generation, so let's breed 2 of these together. When meiosis occurs, every single gamete which is haploid must have one version of every single gene. So every gamete must have a letter R and it must have a letter Y. If the gamete has a capital R, it might have a capital Y with it, or that capital R might have the little y with it instead. So now we've got more possible combinations of gametes. If the recessive letter R is in the gamete that's being produced, then it will either also have a capital Y, or it might have the recessive Y. So there are 4 different possible combinations of these 2 genes, the alleles from these 2 genes in the offspring. Remember, both of our parents have the same genotypes, so this combination can appear in both sets of parents. Now take a couple of minutes to pause the video and make a note of all the possible genotypes from these offspring, and then try and establish what their phenotypes are and the phenotypic ratio is before you move on. Pause your screens again just to check your answers against the diagram on the screen now. Did you have the correct genotypes of the offspring? And have you correctly identified the phenotypes as well? Remember, within the phenotypes, you have to talk about the shape as well as the colour. So pause again and then check your answer. There are 4 possible phenotypes that we're looking for. Either the offspring will have round and yellow seeds, or they will have round and green seeds, or they will have wrinkled and yellow seeds, or they will have wrinkled and green seeds. The next step is to establish what ratio we have of each of those phenotypes, and it literally helps to go through and cross your combinations out one at a time. For example, this offspring is going to have round and yellow. I'm just going to tally it off. This is homozygous dominant for the shape and heterozygous for the colour, but because yellow is dominant, they are still going to have round and yellow, as is that one, as is that one. But go to the next line, round and yellow. This one is round, and because they're homozygous recessive for the colour, it's going to be round and green. So I'll make a note here. And you just keep going through each time, tallying up what your offspring are actually going to be. This one is wrinkled and yellow. Round and yellow, round and green, wrinkled and yellow, and wrinkled and green. So when you write out the ratio, you say there's 9 round yellow to 3 round green to 3 wrinkled yellow to 1 wrinkled green. Pea plants have 14 chromosomes, 7 pairs, 7 from maternal and 7 from paternal side. The reason you get this combination is because the gene for the colour of the seeds is not on the same chromosome as the gene for the shape of the seeds. They are on two separate chromosomes, so they are inherited independently of each other. The round allele might be paired up with the yellow colour, or it might be paired up with the green side. It completely depends on the genotypes, and you are just as likely to inherit either one of them. If both of those two genes were on the same chromosome, then you would get a different pattern of inheritance. Having two different genes on the same chromosome means that they are linked on the autosomal chromosomes. The autosomal chromosomes are the ones where, within each pair, the chromosome from the maternal and the paternal side are the same size and shape as each other, and they have the same genes on both sides. With human beings, there are 22 autosomal pairs of chromosomes. That means there are 22 pairs of chromosomes where the maternal and paternal chromosomes are the same size as each other, the same shape, and they contain the same genes as each other. The 23rd pair is what we call the sex chromosomes, and this is where the size and shape of the maternal and paternal chromosomes might be different to each other. The reason we call them the sex chromosomes is because inheritance of these chromosomes determines whether that individual is going to be male or female. So the human equivalent of the example we just had a look at is you get the dihybrid pattern of inheritance if the two different characteristics you are looking at are on different autosomal pairs of chromosomes. So maybe one of the characteristics was on pair number 1, the other characteristic is on pair number 16. Whatever those different chromosomes are, as long as they're different to each other, then it doesn't matter. They are inherited independent of each other, and you will see dihybrid patterns of inheritance. Where you will get a different ratio to what you expect is if the two characteristics that you're looking at are actually inherited on the same chromosome. So, for example, if characteristic A is on this locus and characteristic B is on this locus, that means that when, during meiosis, the homologous pairs of chromosomes are separated from each other, and then the chromatids are separated from each other, these two characteristics are still going to be on the same chromosome, which means they will be passed on to the next generation together or not at all. Let me show you an example. The inheritance of tail length in these particular types of newts is monohybrid. It is determined by a single gene that decides, is the tail going to be long or is it going to be short? The inheritance of scale colour is also monohybrid. It's determined by a single gene which decides, will the scales be green or will the scales be white? This is an example of autosomal linkage because within one chromosome, the gene for the tail length and the gene for the scale colour is on the same chromosome. For the paternal and maternal chromosomes, you have the same genes on the same locus. It just happens to be that both of these genes are on the same chromosome. If this individual is heterozygous, then maybe they've got the dominant T on one of those chromosomes and the recessive T on the other one. If they are heterozygous for the scale colour as well, maybe they've got the dominant G over here and the recessive G over here. When meiosis happens and reduction division occurs, what you can see as the black and the red chromosome will be separated from each other. And then in the second division, the copies will be separated from each other. But the two dominant alleles are always going to be on the same chromosome, which means you can't inherit the capital T with the lowercase g because that is on a separate chromosome. That limits what the possible allele combinations in the gametes is going to be. So this individual can either pass on capital T and a big G or a lowercase t with a little g. Now, I want you to consider if you have two individuals who are both heterozygous for both of these two genes, how would you put that into a Punnett square? What would be the combination of genotypes in the offspring? And what would be the phenotypic ratio? Pause the video, try and work that out yourselves first before you play the rest to see what it should be. As you can see, the the ratio of phenotypes here is completely different to if it had been a normal dihybrid cross. You have 3 long-tailed green lizards to 1 short-tailed white one. If this had been a normal dihybrid cross between 2 parents who are heterozygous for both characteristics, the ratio would have been 9 to 3 to 3 to 1 again. To check your understanding of the difference between autosomal linkage and normal dihybrid crosses, imagine if this was a dihybrid cross where you have both sets of genes on separate chromosomes. Can you draw out a normal dihybrid cross to show what the phenotypic ratio would have been? A reminder that autosomal linkage means both of the characteristics that you are looking for are coded for on the same chromosome. The only way you are going to get any crossing and inheritance of a dominant T for length of tail and the recessive G for the white scales is if you have crossing over during meiosis, where the chromatids will get tangled up, new chiasmata will form where they've swapped the ends of the chromosomes, and now when the chromosome pairs separate and then the chromatids separate, you can actually get the other possible combinations as well. Note this will only happen if you have random chiasmata forming, if you have random crossing over. Otherwise, you would only get the combination that you would have expected at the beginning.